Don't miss a single minute of this video. I have explained the entire environmental monitoring process in just 5 minutes for you. This video is very helpful to you. If this is first time here, please subscribe my channel and click the bell icon for future videos. Don't skip the video, watch until the end. Welcome to PHARING, a channel of pharmaceuticals. I am your Shaktiver. Environmental monitoring is a systemic process to ensure the cleanliness of drug manufacturing area. The main goal of this monitoring purpose is to protect the product from microorganisms and other non-viable contaminants. Basically, manufacturing areas are classified into several clean room grades like grade A, grade B, C and D. To explain this in a simple way, just consider as a laboratory with multiple rooms. Okay, in the first room we remove our shoes, that is grade D, and next room we wear the aprons like lab coats, shoe covers and mask, that is grade C. Next we enter into the next room for analysis purpose, that is grade B. The same manufacturing area is also classified like raw material storage room, dispensing room and filling station. Here manufacturing area considered as a grade C, but filling area is a highly risk zone, so that is comes under the grade A. So our environmental monitoring peoples monitor these all areas during batch. This process was performed in three ways. One is air sampling and surface monitoring, and the final one personal monitoring. The first method is air sampling, means monitoring the quality of air in manufacturing area. This also performed in two types. One is active air sampling, another one passive air sampling. In active air sampling, the surrounding air is sucked by the mechanical pump through the device. But in passive air sampling, the plate was exposed or kept in a fixed location up to 4 hours to assess the microbial fallout from the surrounding air. Next one is surface monitoring. There are also two types there. One is swab sampling, another one is product plate method. Product is used to monitor the flat surfaces, but swab is used for irregular or non-flat surfaces. This sampling was taken in 5 into 5 centimeter square. This method helped to monitor the contamination on tools which is involved in drug manufacturing process, such as filling syringe, needle, SS table, shelves and other equipment. And the last final method, personal monitoring. As we discussed earlier, filling operations must be performed in grade A zone only. All humans or personnel entering the grade A area for filling activities must and should be perform the personal monitoring. Samples are collected from the personnel at predefined locations such as gloves, forehead, belly or some other locations. This will be varied based on the organizational SOP. In addition, finger tap testing must be performed using a standard 90 mm agar plate at the end of the operation. However, if filling activities are carried out using an isolated system, personal monitoring is not required for operators, but finger tap is required for each and every isolated glove ports. So this is the end of the video. I hope you all have a better idea about the environmental monitoring process in the pharmaceutical industry. So please subscribe my channel and click the bell icon for future videos. If you want to discuss about any topic, means just comment it. We will discuss in next video. Bye.